pleasure to welcome everybody back to session 4, our last session, which is so interestingly titled, What More Do We Need to Know About Microbial Community Dynamics? Our first speaker, Jonathan Eisen, is a professor of the Genome Center at the University of California, Davis. And interestingly, his research focus is on mechanisms of underlying the origin of novelty. But I think, as we've also found, that Jonathan has many talents, and one of them is he's a very big blogger and a microblogger, and apparently he's won awards for his microblogging. That's very interesting in itself, yeah. And anyway, Jonathan is going to talk to us about phylogenetic and phylogenomic approaches to studies of microbial communities.
Thank you, and I guess I have the awkward after lunch talk here, so I'll try not to use the most complicated slide yet used, although I'm not so sure about that. So since I frequently don't get to my last slide, I just want to do like some other people have been doing, acknowledgements at the beginning. And what I'm going to talk about is sort of work funded by a lot of different agencies that has gone on in my lab for about 10, 15 years, including in particular work funded by the Department of Energy, the National Science Foundation, the Gordon and Betty Moore Foundation, and recently Homeland Security, all related to sort of phylogenetic analysis of genomes and metagenomes. And there are a lot of people I will mention, many of the people involved in this, but this is the transdisciplinary type of work. Hurts my head a lot of time to think of all the people involved in some of these projects, but I'll try and acknowledge as many of them as possible. So what I'm going to do is give just a quick introduction to phylogeny and then talk about 3 examples of uses of phylogeny in really in studying microbial communities via DNA sequencing. Phylotpying, functional prediction just a tiny bit because I want to raise the point. It's come up a few times. And then selecting organisms for study. And then I'll end with just a couple of things about future directions. So I assume most people here know what phylogeny is, but just to rediscuss it, phylogeny is a representation of the history of entities. And that could be the history of genes, the history of genomes, the history of species. And in many cases, people have represented this history by a bifurcating tree-like structure. Phylogeny doesn't have to be represented as a bifurcating tree-like structure. We can have reticulation events like recombination and lateral gene transfer. I include all those complexities within the concept of phylogeny. So I'm not trying to discriminate between sort of vertical evolution versus lateral evolution, but really this sort of representation of the history of organisms. I'm also not going to get into sort of the debates about what that exact history is. People are still trying to resolve the evolutionary history of microbes as well as other organisms, and it's a constant area of research. Whatever your sort of belief of the latest model is, in my opinion, if you incorporate phylogenetic approaches in your analysis of genome and metagenome and other data, it can improve what you're doing relative to not trying to incorporate phylogenetic approaches. And what I'm going to do is try and walk you through a couple of examples of this. So the first one I want to talk about is phylotpying, which we've heard either directly or indirectly a lot about at this meeting. And phylotpying I was exposed to as a young, budding scientist in the lab of Colleen Cavanaugh. I was an undergraduate at Harvard and ended up in Colleen's lab, and I spent a year and a half sequencing one 16S ribosomal RNA gene. But I got a paper out of that one 16S ribosomal RNA gene. And the point of sequencing that 16S ribosomal RNA gene, as well as the point, even today in many cases, of ribosomal RNA sequencing, is to try and figure out what the organism is related to where that 16S came from. And the way phylotpying works, this is basically developed by Norm Pace and colleagues. You collect DNA from your sample, you clone out some sequence like ribosomal RNA, you build an evolutionary tree of that sequence. So this is where the phylo part comes in phylotpying, phylogenetic tree of that sequence, and you compare your sort of unknowns to known things that are out there. And this is the tree from our Solomaya velum chemosynthetic symbiont 16S, which, by the way, was accepted 20 years ago tomorrow, I think, my first scientific paper. So, ribosomal RNA phylotpying has been amazing at revolutionizing our understanding of microbes in the world, and I assume most people here appreciate just the vast diversity of things that have been discovered by using phylogenetic trees of ribosomal RNAs from the environment to understand what the organisms are that those ribosomal RNAs came from. I'm not going to go into the whole history of this. What I really want to talk about is 3 challenges that now come up with phylogenetic typing that largely relate to this issue of the cost of sequencing dropping and dropping and dropping and getting easier and easier and easier, accelerating at a rate faster than Moore's law. And with new sequencing machines being announced every 2 or 3 days, not that all of them work, but there are all sorts of cool things coming out there. And so this affects things like PCR amplification of ribosomal RNA sequences. We now have literally trillions of ribosomal RNA sequences to analyze, as opposed to that one that I got a paper out of. It also is really important in terms of revolutionizing metagenomic approaches, and I appreciate what Joe was talking about with metagenomics is not everything about a community, but the cheaper sequencing is, the more data we're going to have from metagenomics. Even though it doesn't tell us everything, we need to analyze that data. And one third challenge is that most of the DNA sequencing technologies that people have been using generate short sequence reads, as opposed to long contigs that are easier to analyze. And so it's sort of obvious that when metagenomic data was generated, you could scan through the metagenomic data to build evolutionary trees of sequences that were in that data. And this is what I did with Craig Venter in the analysis of the Sargasso Sea data. You can scan through the metagenomes, find ribosomal RNA sequences, and build evolutionary trees of those ribosomal RNAs, just like we did with PCR amplified data. The great thing about metagenomic data is we can build phylogenetic trees of other genes that are good phylogenetic markers that we never could really get a good sample of most of these because PCR amplification of protein coding genes across broad diversity does not work very well. So now with metagenomic data, we can look at protein coding genes and compare and contrast the results with those to the results with ribosomal RNA. I've been obsessed with the REC-A gene for a long time, so I always end up working on REC-A, but there are lots of other genes that you can analyze, and we did this in the Sargasso Sea analysis, and if you compare and contrast the results that you get with phylogenetic typing of protein coding genes and assigning those types into phyla, into bins that correspond to the phyla of organisms, you see some interesting patterns. There are some differences between what you get with ribosomal RNA and what you get with protein coding genes. I think a lot of this is due to the differences in copy number. So if you estimate relative abundance of organisms from ribosomal RNA, the copy number of ribosomal RNA varies a lot between taxa, but the copy number of many protein coding genes does not vary a lot between taxa. So the protein coding genes, even though they're not as richly sampled, are probably better markers for estimating relative abundance than ribosomal RNA sequences. So people have been doing this now with metagenomic data in lots of different contexts, and I'm not going to cover all the sort of phylogenetic approaches to metagenomic data, but what I want to talk about is this issue of automation, because I think as we get more and more sequence data, we can't look at trees anymore. We can't look at sequence alignments. We can't even handle all the data at all, but we certainly need to automate everything. And so there are multiple strategies to trying to automate phylogenetic typing of ribosomal RNA or metagenomic data. And one of them has been to use the BLAST program or analogs of the BLAST program, which basically looks at sequence similarity of your sequence to sequences in databases. This is not the best approach to analyzing data. Percent similarity or other measures of similarity are not a good indicator of evolutionary relatedness and can produce misleading patterns about sort of the taxonomy and other parts of information that you want to analyze. There are also approaches that look at compositional and word frequencies. Now, both of these approaches are very fast, so you can generate lots of results very rapidly, and that can be an advantage in many cases. But phylogenetic analysis is generally better than most of these approaches. And the challenge is, how do you implement phylogenetic analysis on a massive scale? And so what I'm going to do is just give you sort of 4 examples of some of the issues related to implementing automated phylogenetic analysis on a large scale. So you can scan through the data and say, I'm going to take each individual sequence, each individual new thing that I get, and build an evolutionary tree of it relative to known things. So in essence, each sequence is an island in and of itself. So we've done this with a variety of tools. We built an automated ribosomal RNA tool called STAP, which goes through and takes a reference alignment of known ribosomal RNAs, and then for each new sequence, aligns your new sequence to that and builds an evolutionary tree in a completely automated manner, and then can scan through the tree to look at the taxonomy results from the tree. We also built a tool that will do this with protein coding genes called Amphora, so it can automatically scan through metagenomic data, find homologs of particular protein families, build an alignment of them, build an evolutionary tree of them, and if you have a good reference alignment from known organisms, you can identify what those protein coding genes come from in your environmental sample. Now, having a good reference sort of database is challenging for protein coding sequences, whereas we have now trillions of ribosomal RNA sequences and tens of thousands of complete ribosomal RNA sequences. We don't have good databases of protein coding sequences. All of the good data is now coming from genome sequencing projects. So whatever has been sequenced in terms of genomes is basically our source of protein coding genes for building these evolutionary trees. And so you build a reference tree from the genomes, you take your new data, stream it against the reference tree, build a new tree with that, and assign your new sequence to somewhere compared to the reference data. And we've done this with this Amphora. It can allow you to stream through massive metagenomic data sets and do taxonomic assignments for a suite of protein coding genes, just again like you would do with ribosomal RNA sequences. And again, I think this is very advantageous in particular because of the copy number variation with ribosomal RNA. I'll skip over this. We've shown that it's better than sort of similarity-based approaches. So this is analyzing each individual sequence on its own. But of course, when you sequence from a new environment, you also want to compare the new sequences to each other. You don't want to compare each one individually to the reference data. And so there are a lot of methods that people have been trying to develop to build evolutionary trees of all the new sequences compared to each other. Does this keep kicking out? Or okay. One of the challenges with this is when you have metagenomic data in particular, the new sequences that you get might not correspond to the entire length of the reference sequences that you're analyzing. So you might have an alignment that looks like this. One solution to this is to just trim the alignment and only pick out regions from the metagenomic data that overlap with everything in your reference database. And so we and other people have built methods to do this, to go through, take all the new sequences, align them to the reference data, and now chop off a core region that sort of everything has a component of, and build an evolutionary tree of that. I did this by hand to analyze the Sargasso data with ribosomal RNAs and a variety of other sequences, including RECA, et cetera. All of that was done by hand. It's much better to automate this. So again, we've added a step in this for this ribosomal RNA pipeline. Now, there are many other tools to do this with ribosomal RNA, so Chime, Mother, a lot of tools out there will build alignments for you with ribosomal RNA and help you build trees of everything. Usually these work best when you have all the sequences overlap with each other. So the challenge, again, is what you do in cases where the sequences don't overlap with each other completely, as you would get with metagenomic data. Again, so I did this by hand, but we've developed methods that can allow you to do this for protein coding sequences and compare them all to each other. So in the Sargasso data, you can't really see it, but in red were sequences from the Sargasso, I guess you can see it, from the Sargasso Sea, and in black were sequences that were from genomes. So you can see how those new sequences relate to each other in addition to how they relate to the reference data. So this is sort of constraining yourself to this core region of the sequence alignment. But there are methods available that people have used primarily in analysis of morphological data or of express sequence tag data, EST data, where you can build an evolutionary tree of sequences that don't overlap with each other at all. So if you have good reference data and you have a sequence that matches the left hand of the reference data and a sequence that matches the right hand of the reference data, that's sort of like if you went to an archaeological or paleontological dig and you had a femur bone from one organism and maybe some teeth from another. And you can figure out, in essence, whether or not they might have come from the same organism by comparing them to references. So you can do the same thing with sequences. And the latest sort of in phylogenetic analysis of metagenomic data has been to try and build methods that will build evolutionary trees even when sequences don't overlap with each other at all, by using the reference sequences as your anchor. And so we've developed a few tools in my lab in collaboration with Katie Pollard and Jessica Green. One is called PhyloOTU, which identifies OTUs using this approach. We have another one called PhyloSift, which is like the new version of Amphora that will do this for protein coding genes. There's a great method called pPlacer that we've integrated into PhyloSift from Eric Mattson. Eric Mattson's now at the Hutch in Seattle that has been developed to do this exact type of thing. Again, you can build trees for sequences even if they don't overlap with each other. So the final frontier in this is to try and build trees even with different genes when they don't overlap with each other. And so we did a little test case of this in collaboration with Stephen Kemble and Jessica Green in Oregon, where we basically took all of the genes that we'd been analyzing in that Amphora package, found homologs of those, and now built a reference tree of a concatenation of all of those sequences. And then for each sequence that matched any of those individual sequences, different protein families in the environmental data, we can build an evolutionary tree that fits them relative to this anchor of the concatenated alignment of all sequences. So in the long run, I think this is what we're going to want to do with environmental data is, for all genomes, build up a reference tree of all the gene families in those genomes, and then anchor environmental data to that reference tree, and you can figure out much more precisely where those sequences came from, even if they're not part of a traditional sort of evolutionary marker gene family. This is the result of this. So Steve Kemble did this analysis not to do phylotyping, but because he wanted an evolutionary tree to do what's called phylogenetic ecology. So many people are probably familiar with UNIFRAC analysis to compare sort of the diversity of communities by their overlap and the amount of phylogenetic sort of tree that they cover from the two communities, the unique fraction of the evolutionary tree. That's an approach which could generally be called phylogenetic ecology. And Steve Kemble was really interested in comparing phylogenetic diversity between communities with metagenomic data. And the reason he wanted to concatenate all of these different genes was we didn't have enough sequences from individual genes to have enough signal. But if you have 100 genes that you can analyze at once, from across the genomes, you can build up enough signal to ask questions about beta diversity and et cetera in ecological communities. Yeah?
What do you think is the meaning of the especially long branches that you have, let's say 5 or 6 of?
Yeah, so the question is, what is the meaning of the especially long branches? In this case, I think it is taxa that actually evolve rapidly. So in the reference data here, we have some organisms like endosymbionts, mycoplasmas, et cetera, that every one of their genes evolves on a long branch. You can get artifacts in some of these cases where you have too small of a fragment, and the phylogenetic methods just get confused by that and give you a really long branch length. I don't think that's the case here. So I think most of the cases here are where taxa are known to evolve more rapidly, and the branch length is in essence a representation of evolutionary rate. Yeah. The colors were different. Sorry, I didn't want to go into the ecological detail here. So what Steve analyzed was Ed DeLong's Hawaii Ocean Time Series data, reanalyzed that, and the colors correspond to different samples from Ed's data. And so what he was asking basically was primarily whether or not phylogenetic approaches to calculating beta diversity gave different answers than taxonomic approaches to calculating beta diversity, where you just count organisms as opposed to comparing the phylogenetic relatedness of organisms. So again, it's analogous to UNIFRAC, but now you can do it with metagenomic data, not just with ribosomal RNA data. Another thing that I'm very interested in and have been interested in for a while is to look for novel lineages in metagenomic data. And we wanted to do this a long time ago, and we ran into some sort of bioinformatic roadblocks. So what I really wanted to do was scan through metagenomic data to find whether or not there was evidence for a fourth branch in the tree, or something to that effect, things that were really phylogenetically novel compared to other sequences. And we had this problem, which was if we did this for any type of data set like RecA at the time, we had something like 10,000 rec-A sequences from bacteria, 200 from archaea and 200 from eukaryotes. And at the time, building evolutionary trees of 10,000, 10,000 sequences or more were challenging. So we wanted to sort of remove many of the bacterial sequences and only analyze a couple of them, as opposed to analyzing the 10,000 bacterial sequences. And what we did was develop a method that's an analog of something called Lek clustering, where you take sequences, you group them together in essence into subfamilies, and you can identify sets of sequences that are related to each other really rapidly. And that's what we use to flag different subgroups in these massive data sets of 10, 15,000, 100,000 sequences. And when we do this for rec-A or RNA polymerase or other protein family sequences and scan through metagenomic data, you find lineages that don't group into any known current lineages of organisms. Now, these easily could be coming from viruses that are out there in the environment. They could be new paralogs of rec-A that are previously uncharacterized, or they could be coming from phylogenetically very novel lineages that are out there in the environment. And the way to find phylogenetically novel lineages is to build evolutionary trees of all the sequences that you can get from environmental samples. And I don't know if Jill's going to talk about this. I know Jill has done this and found novel archaeal lineages, for example, in metagenomic data within the archaea. What we were looking for here was basically, is there anything that can show up between bacteria, archaea, and eukaryotes? My guess is these are not, cellular organisms. They're just DNA sequences, and they're probably from viruses or something to that effect. But again, phylogenetic approaches are the way to scan through this type of data to find novel lineages. I'm not going to talk about this, but phylotpying is also very useful for binning metagenomic data, for trying to pull things together into one group that corresponds to a particular organism. We've done this previously with endosymbionts, for example, but you can use it with any type of data. So I want to shift gears. I think phylotpying is a really important area, and we're doing a lot of research on it, as are many others. And I want to shift gears just quickly to talk about a few other uses of phylogenetic analysis for studying microbial communities or microbes. And one relates to functional diversity and functional prediction. And I've spent a lot of time working on this over the years, and I've been very interested in basically, when you have new sequence data, how do you make a prediction of the function of that gene? We've talked about this a little bit at this meeting with the examples of the cytochrome oxidases and a few other examples here. How do you make a robust prediction for a sequence of a gene? And I think just like analyzing ribosomal RNA sequences to understand an organism by building an evolutionary tree of those sequences, you should build an evolutionary tree of protein family sequences in order to understand the functional diversity in a family. And I developed an approach that I originally called phylogenomics to do this many years ago, and it's basically you take a sequence, you compare it to its homologs, you build an evolutionary tree of that sequence and its homologs, you overlay experimentally determined functions onto the tree, and then you use character state reconstruction methods to predict the functions of unknowns. Sound familiar, anybody? It's phylotpying. I copied this completely from the ribosomal RNA world, but you can apply it to functions as opposed to organisms. And it's a very powerful tool in predicting functions for uncharacterized genes. Again, placing them in their phylogenetic context is incredibly powerful. I think in the interest of time, I won't go into the multiple examples that I have of this. I'd be happy to talk to people about this. This is routinely used now in many genome analysis projects to build evolutionary trees of various sequences. Again, you could do it with whatever sequences you want from environmental data as well as from sequenced genomes. The latest thing in functional prediction, which I think is really interesting, is to use these non-homology functional prediction methods, which look at things like distribution of patterns of genes across organisms. You can also use distribution patterns of genes across environments to try and help you make predictions of functions of genes. This has been done in a variety of metagenomic projects. Exactly how you group genes and analyze the correlation between the distribution pattern of a gene and the taxa present in a sample and the metadata of the sample is still sort of a work in progress. We've been collaborating as part, Simon left, but as part of a project that Simon Levin was in charge of, this DARPA Fundamental Laws of Biology project with people from Simon's lab to try and figure out methods to analyze gene family distribution and correlate it with taxa or with metadata from environmental samples. So the last thing I want to talk about is this issue of selecting organisms for study. So a different use of phylogenetics is to try and understand what we've known about in the diversity of life. And so if you go through the ribosomal RNA tree of life, many people probably are sick of these slides if they've seen me talk about it before. There are many different lineages of bacteria. In 2002, when we first sort of noted this ourselves, most of the genomes came from 3 phyla. That's still basically true. There were some genome sequences available from other lineages, but most lineages were poorly sampled. The same trend is true for eukaryotes, true for archaea, and true for viruses. So we had a project originally when I was at Tiger to sequence 8 representatives of novel phyla for which genomes were not available that was funded by NSF. More recently, I've been coordinating a project called the Genomic Encyclopedia of Bacteria and Archaea at the DOE Joint Genome Institute, where we've been really filling in the tree of bacteria and archaea of cultured organisms with representative genome sequences. And this is one of these massive projects with hundreds of people involved, and it's been this amazing collaboration with the DOE JGI and the DSMZ culture collection. And what we did is basically go through the tree of life, selected genomes to sequence by their phylogenetic novelty, and then ask questions about whether or not phylogeny ended up being a useful guide in selecting genome sequences. And we've shown now about 5 or 6 areas where phylogenetic sampling has improved our analysis of genome or metagenome data. So one is in functional predictions of genes. Another is in discovery of genetic diversity. So a phylogenetically novel organism, if all else is equal, is more likely to have new protein families than a phylogenetically not novel organism. We also showed that these phylogenetically novel organisms could help you analyze metagenomic data by providing more reference data across the tree in essence. But when we did this, there was very little benefit to analyzing the metagenomic data from the first 50 or even 100 genomes that we sequenced from this Genomic Encyclopedia project. And the reason for this is the last thing I want to talk about, which is we need to adapt many of the methods that we're doing to improve our ability to make use of this phylogenetically diverse data. So we need to design new phylogenetic methods, new metagenomic methods to take into account this environmental data. And I've been involved in this great collaboration with Jessica Green and Katie Pollard and Martin Wu to try and develop methods to take advantage of this. It just ended. We called it Eisen. I guess this is my most complex slide. This is one of the products of that, which is a tree of 2,500 genomes that Jenna Morgan and Aaron Darling in my lab generated. We've been building new protein family markers from all of these genomes. So we can improve that amphora pipeline by having hundreds to thousands of new phylogenetic markers to use to scan through metagenomic data. And the last thing I want to leave you with is the reason why I think this project did not really help analyze metagenomic data, which is we haven't even begun to scratch the surface of microbial diversity in terms of reference genome data. So if you go through the tree of life and you count the branch length in the tree, it's something called PD, or phylogenetic diversity, and you sum up the total length of the branches for all the genomes sequenced before our project, came to 25 units. Each genome that we added a lot of new units of phylogenetic diversity. It better have, because that's how we picked them. If you go through cultured organisms that are known and described, so the 8,000 or so described bacteria and archaea, we would need about 1,000 genomes to capture half of that diversity. This will be done in the next year or two. However, if you go through all the environmental data, we would need about 10,000 genomes to capture half of the diversity known 5 years ago in full-length ribosomal RNA sequences. So the vast majority of genomic diversity out there is uncaptured in most studies of cultured organisms. I think the solution to this is to do genomes of uncultured organisms, either via single-cell capture or, as I think you'll hear from Jill and other people, metagenomic sequencing and assembly of those metagenomes can generate genome data from uncultured organisms. And by doing that, we will really fill in the tree, and that will enable all sorts of different uses of phylogeny